Here we are going to start another bacterial disease which is caused by a bacteria and having a drastic effect on the host because of this disease. And this disease which is caused by this bacteria, that disease we call it as a pulmonary tuberculosis. And this disease is caused by bacteria Mycobacterium tuberculosis, and this bacteria belongs to a genus. That genus we call it as a mycobacteria. You can observe here in this slide, these are the bacterial growth which are affecting this. These are the bacteria which are affecting the lungs of that host, and because of the affection of these lungs, they affecting these lungs very bad. And in last stages of this infection, you are able to observe while that person having a cough, in that cup you are able to observe readily the presence of blood sample. So this is one of the drastic infection caused by this bacteria Mycobacterium tuberculosis, which belongs to a genus. That genus we call Mycobacterium, and this Mycobacterium genus contains those group of bacteria which are showing slender rods, and sometimes this bacteria shows filamentous branching which looks like the fungal mycelium, which resembles to the fungal mycelium. And in liquid cultures, these bacteria they form mold-like pellicle on the surface of that medium. So that's why the name Mycobacterium is given to this group of bacteria, means which look like a fungus. So fungus-like bacteria. This is the name which is derived from the Mycobacterium. Mycobacteria, it is the name which is derived from fungus-like bacteria. And these bacteria, they don't stain readily, but once they get stained, they resist decolorization with dilute mineral acid. So that's why this Mycobacterium group, they are also called as acid-fast bacteria or AFB. This group of bacteria, they are aerobic, non-motile, non-capsulated, non-spore forming, and this bacteria also shows another characteristic. Another characteristic is that this bacterial growth is very slow. So in order to grow this bacteria, longer duration is required. And this group of genus, this Mycobacteria genus, it includes various bacteria which causes various kinds of infections. Now this group includes obligate parasites, opportunistic pathogens, and some saprophytes. So this is the genus. So this is the genus from which a group of bacteria belongs, and that group of bacteria is responsible for causing pulmonary tuberculosis disease in human beings. And this is one of the dangerous disease caused by this group of bacteria. Now here, this group of bacteria, you can observe this is the growth of Mycobacterium, which looks like the mycelial growth. And that's why this group of bacteria, they are called as Mycobacteria, fungus-like bacteria. And because of that, this name Mycobacterium, it is derived. Now the first member of this genus was identified as Liparibacilli, which was discovered by the scientist Hans in 1868. Later on, in 1882, Robert Koch isolated the mammalian tubercle bacilli and proved its causative role in causing tuberculosis by using the Koch postulates. Now, tuberculosis bacilli in humans is subsequently shown to be caused by two types of bacilli, the human and the bovine type. And the human type of bacteria, human type of mycobacteria, tuberculosis bacteria, that we call it as a Mycobacterium tuberculosis. And those bacteria which are present in animals and then later on causes the infection to human beings, such bacteria, they are grouped under the Mycobacterium bovis. And so that we call it as a bovine type of mycobacteria, which causes the infections to humans. And those mycobacteria which causes infection to human beings directly, such bacteria we call it as a Mycobacterium tuberculosis. And those bacteria which are saprophytes without causing any infections, and such type of bacteria, such type of mycobacteria, they are called as atypical mycobacteria. Okay, so these are the two species of mycobacteria which are responsible for causing pulmonary tuberculosis in human beings. Whether it is originated from human, then it will be called as a Mycobacterium tuberculosis. If the infection originated from the bovine type of mycobacteria, then we call it as a Mycobacterium bovis. So here we have to see the infection caused by Mycobacterium tuberculosis, and then this pulmonary tuberculosis which occurs in human being is caused by Mycobacterium tuberculosis. Now here we are going to see the etiological characteristic of this bacteria, Mycobacterium tuberculosis, which is responsible for causing pulmonary tuberculosis. Firstly, the morphology of this bacteria. This Mycobacterium tuberculosis bacilli, it is straight or slightly curved rod and about 3 micrometer to 0.3 micrometer in size. They may occurs singly, in pairs, or in a small clumps. The size depends upon the growth conditions. Sometimes they form long filamentous, club-shaped and branching forms. Okay, this Mycobacterium tuberculosis, the growth of this bacteria, the size of this bacteria depends upon the growth condition and in which growth condition this bacteria is growing. According to that, the various forms of this bacterial colony or growth we are going to observe. Here, long filamentous club-shaped branching forms, they are also seen in the Mycobacterium tuberculosis. While in case of Mycobacterium bovis, the size of the bacteria, it is a straighter, shorter and stouter, but it is a rod one. Tubercular bacilli have been described as a gram-positive, but strictly speaking, this is not the correct, because during the gram staining process, we are going to use mordant, then decolorization step, and lastly the last stain, that is saffron. But in this case, once this bacteria, they get stained by using basic dyes, they are not decolorized, even with the help of acid or alcohol. So we can not call this bacteria as a gram positive, but we can call this bacteria as an acid fast bacteria. Morphologically, this Mycobacterium tuberculosis and the Mycobacterium bovis, they are rod in shape. Now, a special type of staining method is there. With that method, we called as a Zell-Nielsen method, or that is acid fast staining method. When we stain this bacteria by using carbol fuchsin, using the method of Zell's and Nielsen method, and similarly we can stain this bacteria by using some fluorescent dyes, aramine or rhodamine. These are the fluorescent dyes that can be used in order to stain this bacteria and readily observed under the microscope. Now, in Zell's-Nielsen method, this bacteria can reach the decolorization by using 20% of sulfuric acid and absolute alcohol for 10 minutes. So that's why the name acid fast is given. Acid fastness means they are, as even by using the acid, we are not able to decolorize this bacteria. So the name acid fast is given. And the acid fastness of this bacteria, it is because of the presence of in this bacillus a unsaponifiable wax, that is mycolic acid. It's because of the presence of this mycolic acid wax, this bacteria reaches the decolorization even with the help of alcohol and acid. Okay, so because of this, they are not able to decolorize easily with the help of acid and alcohol. And that's why these bacteria, they are called as acid fast bacteria. You can observe here, these pink colored bacteria, they are the acid fast bacteria, which is the stained after, which is the sputum sample which is stained there by using this acid fast staining method. And you are readily going to observe here the presence of this pink colored bacteria indicates that the person's sputum sample is positive for the pulmonary tuberculosis infection. You can see here all these figures, all these photos, they are of acid fast staining method of sputum sample. This is you can here readily observe the presence of rod-shaped bacteria. Same is here. This is also the positive phase. Then the cultural characteristic of this bacteria. This bacilli grow very slowly, and the generation time of this bacteria is about 14 to 15 hours in vitro. In laboratory conditions, it requires near about 14 to 15 hours for one bacteria to grow into two bacteria. So the growth rate of this bacteria is very slow. It appears that the colonies of this bacteria will be developed. For the development of the colonies of this bacteria requires near about 2 weeks, but may sometimes take up to 8 weeks. So it's because of the slow growth rate of this bacteria. This development of the colonies takes a lot of time. As you know that in the E. coli bacteria, it can divide and produce two E. coli bacteria within 20 minutes. But this Mycobacterium tuberculosis and bovis, these bacteria, they are the slow growers. Here, the generation time for one bacteria to another, it is 14 to 15 hours. And that's why the colonies developed on the medium, they took 2 weeks to may sometimes up to 8 weeks. The optimum temperature which is required for the growth of this bacteria, it is 37 degrees Celsius temperature. This bacteria doesn't grow below temperature range 25 and above temperature range 40. So the optimum temperature required for the growth of this Mycobacterium tuberculosis and the bovis is 37 degrees Celsius. Similarly, the optimum pH is also required for the growth of this bacteria, which is 6.4 to 7. This is the optimum pH which is required for the growth of this bacteria. Now this Mycobacterium tuberculosis is an obligate aerobe, while Mycobacterium bovis is microaerophilic on primary isolation, but later on becomes aerobic on subculture. So this Mycobacterium tuberculosis requires strict aerobic condition for their growth. Without oxygen, they are not able to survive there or grow there. And this Mycobacterium bovis, it is earlierly microaerophilic when we isolate firstly this bacteria, and later on subculturing it becomes aerophilic again. Mycobacterium tuberculosis grow luxuriantly in culture as compared to the Mycobacterium bovis, which is grows sparsely. Means grow very less growth is there. And because of these two characteristics, these bacteria, they are also called as eugonic and dysogonic type of bacteria. Then, in another characteristic which is shown in this cultural characteristic is when we add 0.5% of glycerol in the medium, it improves the growth of Mycobacterium tuberculosis. But this 0.5% of glycerol affects the growth of Mycobacterium bovis. So we can selectively allow the growth of these two bacteria. If we are going to add 0.5% in glycerol in the medium, then we are going to allow only the pathogen Mycobacterium tuberculosis to grow there. We can easily exclude this Mycobacterium bovis bacteria. Now, sodium pyruvate, it helps the growth of both the bacteria, both the type of bacteria. Suppose we have added sodium pyruvate in the medium, both the kinds of this bacteria can readily grow on this medium. This human tuberculosis bacilli, they don't grow in the presence of a compound that is para-nitrobenzoic acid. This para-nitrobenzoic acid, it inhibits the growth of Mycobacterium tuberculosis. And this is the another characteristic which can be used selectively for the isolation purpose of this bacteria. If we are adding para-nitrobenzoic acid, benzoic acid in the medium, then Mycobacterium tuberculosis will not grow. The other strains will be easily growing on this medium. This tuberculosis bacilli, they don't have exact growth requirements, but they are highly susceptible to the trace amount of toxic substances like fatty acid. If we have added the fatty acid in the medium, then this tuberculosis bacilli, Mycobacterium tuberculosis bacilli, they are not able to grow there. So this fatty acid, they are toxic to them. This is another cultural characteristic of this Mycobacterium tuberculosis. Now, special kinds of solid mediums are available for the growth, for the isolation and the growth of this bacteria. But these solid mediums, they contain some of the selective components, such as egg. Some solid medium also contains blood, serum, potato. These are the various scientists which they have developed this medium. So that's why these names are given in the bracket. Now, a special type of solid medium is routinely used for the growth of this bacteria. That medium is known as a Lowenstein-Jensen medium, and this medium is without starch, is used, and it is recommended by International Union Against Tuberculosis. This is the union which fought against this tuberculosis disease. So this medium is recommended by this union in order to grow in laboratories. So most commonly this Lowenstein-Jensen medium is commonly used for the growth of for the isolation and the growth of this Mycobacterium tuberculosis and Mycobacterium bovis bacteria. Now this medium contains coagulated hen's egg, mineral salt solution, asparagine and malchate green. Here the malchate green, it acts as a selective agent which is inhibiting the other types of bacteria. It only allows the growth of Mycobacterium group of bacteria. Now on solid media, this Mycobacterium tuberculosis forms dry, rough, raised, irregular colonies with wrinkled surface. And earlier, the colonies of this bacteria on this Lowenstein-Jensen medium, they are creamy white. But as we continue to incubate this bacteria on the medium, the colony color becomes changed. That becomes yellowish or buff colored after prolonged incubation. Now this is the colonies produced by this bacteria. They are firm. They are not easily emulsified. Means we cannot easily separate the colony from the medium. It is so hard. And the Mycobacterium bovis colonies, In comparison to this Mycobacterium tuberculosis, they are flat, smooth, moist, white, and break up easily when touched. So we can differentiate between the Mycobacterium tuberculosis colonies and the Mycobacterium bovis colonies. You can observe here, these are the colony morphologies which is developed on the Lowenstein-Jensen medium, where you can able to observe the wrinkle-shaped, irregular-shaped colonies of Mycobacterium bovis, and these are the colonies of Mycobacterium tuberculosis colonies. Is this one, same is here, and this is the colonies of Mycobacterium bovis. You can also observe colony morphology in another photo. Here, these are the wrinkle-shaped colonies of Mycobacterium tuberculosis. Okay, and this bacteria have grown on the Lowenstein-Jensen medium, and on this medium they produce this type of colonies, and this medium is very selective medium. It only allows the growth of this bacteria, but the growth of these bacteria on this medium, it requires lot of time to develop. Here, it may require 2 weeks or maximally 9 weeks to grow on this medium. Now, when you grow this bacteria on liquid medium, without any dispersing agent, the growth of this bacteria begins at the bottom, which creeps up the sides and forms a prominent surface pellicle, and which may extend along the sides above the medium. Here, this bacteria also shows characteristic growth on the lipid medium. Firstly, the growth of this bacteria begins at the bottom, which then creeps from the side of the test tube, and then they forms the pellicle on the surface of that lipid medium. When we use diffuse medium, which contains, take one example is given here, Dubose medium, which contains twin 80. This is the detergent which is used here. Here, when we grow this bacteria on this Dubose medium, it forms diffuse growth, and virulent strains tend to form a long serpentine cord in the liquid medium. This is another characteristic which is shown by this bacteria in the liquid medium. And now another characteristic, resistance characteristic of this bacteria. This mycobacteria, they are not specifically heat resistant. They can be easily killed at 60 degrees Celsius temperature for 15 to 20 minutes. Now there are several factors are there which affects the growth of this bacteria. Now this culture may be killed by exposure to the sunlight for 2 hours. 2 hours sunlight exposure, this bacteria will be dead there. This bacilli remains in the sputum sample, remain alive for 20 to 30 hours. While this bacilli remain in the droplet nuclei, may remain alive up to 8 to 10 days under suitable conditions. And this bacterial culture, when we isolate them, they remain viable at room temperature for 6 to 8 months. And it may be stored for up to 2 years at minus 20 degrees Celsius temperature. So, this bacteria can remain alive in the droplet nuclei, which are produced because of the sneezing. Because of the sneezing process, whatever the droplet they comes out from the sneezing process, they remain float in the air. And this droplet nuclei, this Mycobacterium tuberculosis and bovis bacteria, they can remain viable for 8 to 10 days under suitable conditions. If high sunlight is not there, under such conditions, this bacteria, they can survive in this droplet nuclei for 8 to 10 days. This is very important because this acts as a source for the infections to the healthy person. Now this tuberculosis bacilli, they can be relatively resistant to chemical disinfectants. They can survive exposure to 0.5 to 5% phenol, 15% sulfuric acid, 3% nitric acid, 5% oxalic acid, and 4% sodium hydroxide. So this much concentration of these compounds, this bacteria can survive. But these bacteria, they are highly sensitive to the formaldehyde and glutaraldehyde. These are the two chemical components to which these bacteria, they are highly sensitive. These bacteria can be destroyed by a tincture of iodine in a 5 minutes and by 80% ethanol in 2 to 10 minutes. So glutaraldehyde and formaldehyde, they are playing important role in order to cause disinfection of the surfaces where this pathogen may present. Here, ethanol is also suitable disinfectant for skin, gloves, and clinical thermometers, which are used in hospitals. You can observe these are the liquid medium where the growth of this bacteria begins at the bottom, then it comes to the surface of that bacteria, and there on the surface they forms the pellicle. You can see here, while this person is coughing or sneezing, it releases this much amount of droplet.
स्लाइड दिसत नाहीये.
काय?
स्लाइड दिसत नाहीये.
Okay, just a minute. आता दिसते? स्लाइड दिसते का आता?
नाही सर.
नाही?
हा सर दिसायली. हो सर दिसली.
Okay. So this is a person who is coughing and sneezing, and it releasing large amount of droplet nuclei in the environment. And if that person is suffering from the tuberculosis disease, then it has released large amount of this bacteria into the environment. And because of that, and as you know that this bacteria can survive in this droplet nucleus for up to 8 to 10 days under suitable conditions, then it may act as an important source to begin a new infection in a healthy person. Okay. Now there are certain biochemical reactions which are used to differentiate between the Mycobacterium tuberculosis and the Mycobacterium bovis and Mycobacterium, atypical Mycobacterium. The first test is the Nicin's test. This Nicin's test is positive for Mycobacterium tuberculosis, where this bacteria forms niacin when it is grown in a medium containing egg. And to that egg medium, if we are going to add 10% cyanobromide and 4% aniline in 96% ethanol is added to that suspension of this bacterial growth, then there is a development of canary yellow color, which indicates the positive niacin positive test, and this test is positive for Mycobacterium tuberculosis, while other Mycobacterium strains or species, they are not capable of producing this niacin. You can see here this canary yellow color, presence of this canary yellow color means this test is positive for Mycobacterium tuberculosis bacteria, while this is the negative test where the Mycobacterium bovis, it is negative. And this is also another positive test for the niacin test. Then another biochemical test, aryl sulfur test. This test is positive only for atypical bacteria. Atypical bacteria means those bacteria which are saprophytic, and they came to that source, that host from a saprophytic conditions. Then we can easily differentiate between saprophytic bacteria and the pathogenic bacteria. Here, this aryl sulfur test, it is test positive for atypical bacteria only, while this pathogenic bacteria, they are not able to show this test positive. Here, simply these bacteria, they are grown in a medium containing 0.001 molar tripotassium phenolphthalein diphosphate. Here in this medium, these bacteria, they are allowed to grow there, and after growth there is an addition of 2 normal NaOH which is added there drop by drop to that culture medium. A pink color indicates positive reaction. This is the positive reaction, while this is the negative, and this is the control again. So this aryl test is positive for aryl sulfatase test. It is positive for atypical bacteria only, while the pathogenic bacteria, they are not capable of showing this test positive. It is only shown positive by atypical bacteria. Then another biochemical reaction, that is neutral rate test. This test is positive for virulent strains of tubercular bacilli because they are capable of binding to the neutral rate in alkaline buffer solution. Means this bacteria binds to the neutral rate stain and they forms the another kind of nature there. While the avirulent strains, they are not unable to show this test positive. Now another important test is that catalase peroxidase test. This test helps to differentiate between tubercular bacilli from atypical bacilli and provide an indication also the sensitivity of these strains to the isoniazid, that is an antibiotic. If this antibiotic is used in the treatment of this tuberculosis patient, so it also indicates whether that strain which we have isolated from that patient's sample specimens, that is pathogenic or not, and again whether it is sensitive to this antibiotic or not. Now mostly this atypical mycobacterial strains, they are strongly catalase positive. So this catalase peroxidase test, it is used to differentiate between pathogenic and non-pathogenic bacteria. Here tuberculosis bacilli, both tuberculosis, Mycobacterium tuberculosis and Mycobacterium bovis, these two bacteria, they show this test, and atypical bacteria, we are going to easily differentiate between this pathogenic and non-pathogenic bacterial strains. Here the atypical mycobacterial strains, they are strongly catalase test positive, while the tuberculosis bacilli, they are only weak positive in comparison to the atypical bacteria. And on the other hand, tuberculosis bacilli, they are strongly peroxidase positive, while the atypical bacteria, they are negative for this test. So that this catalase peroxidase test is used to differentiate between atypical and the pathogenic bacteria. Now catalase and peroxidase test activities are lost. Both these activities of this bacteria, they have been lost when this bacteria becomes resistant to isoniazid antibiotic. So we are easily to get the information about the sensitivity of this bacteria against the antibiotic which is used for the treatment of pulmonary tuberculosis. So we can easily get this information whether this is sensitive to isoniazid or not. Now catalase test is negative for the tuberculosis bacillus strain, are not virulent to genetics. Now in order to carry out this catalase peroxidase test, a mixture of equal volume of hydrogen peroxide and which is taken in 30 ml and 0.2% of catechol, they are taken in a distilled water, and to that 5 ml of test culture is added there and allowed to stand for few minutes. If presence of effervescence indicates the catalase test positive and browning indicates the peroxidase test positive. And this is the neutral rate reaction. You can see here neutral rate is added to this test tube, and this neutral rate is used by the bacteria and fixed there. And while this is the negative test for negative neutral test. This is the catalase test positive. You can see the presence of effervescence in that medium where is here H2O2 is taken. After that the bacterial sample is added if there is a presence of effervescence is there, then this catalase test is positive, and this catalase test is positive for a typical bacteria only. And while this catalase test is shown negative or weak by this pathogenic bacteria. This is the peroxidase test. Presence of this browning color indicates the peroxidase test positive test, while this is the negative test. And this positive test is peroxidase test positive is given by the pathogenic tuberculosis bacilli. And if both these tests they are negative, it indicates that this isolated bacterial mycobacterium strain is resistant to the isoniazid antibiotic. If both the tests they are shown negative at that time. Now another test is the amidase test, and this amidase test is shown positive by the pathogenic mycobacteriums. And here the ability of this bacteria is checked to split the amidase, and it is useful for the differentiation of mycobacteria. Here 5 different types of amidase they have been used in this test medium: acetamide, benzamide, carbamide, nicotinamide, and pyrazinamide. These are the various amides they have been used in the test medium. Here 0.00165 ml molar solution of these amides they have been incubated with the bacterial suspension at 37 degrees Celsius temperature, and to that 0.1 ml of imidazole 4 hydrated, 1 ml of phenol solution, and 0.5 ml of hypochlorite solution they added. And these tubes they are placed in the boiling water for 20 minutes. A blue color indicates the positive amidase test. Means amidase enzyme is produced which split these 5 different types of amidase into another compounds which reacts with these reagents and forms the blue color. And here you can see here, this is the blue colorations which is formed there, and this is the negative test shown by the bacteria. And this test is positive for pathogenic one. Another test is that nitrate reduction test, and this nitrate reduction test is shown positive by Mycobacterium tuberculosis but shown negative by Mycobacterium bovis. Here Mycobacterium tuberculosis is able to produce a nitroreductase enzyme which reduces nitrate to nitrite, and this is detected by adding sulfonylamide and N-naphthalene ethylene diamine dihydrochloride. These reagents they have been added to the test culture medium, and presence the pink color or red color indicates the positive test, and this test is shown positive by Mycobacterium tuberculosis, while the Mycobacterium bovis it shows this test negative.